Hey Mustangs, in this video we're going to take a closer look at how enzymes work, so exactly how they lower activation energy in order to control or speed up a chemical reaction. Okay, so first thing, let's go ahead and take a look at enzymes and substrates. Okay, so remember, enzyme is a special type of protein that can control or speed up a chemical reaction, and it does that by lowering the amount of energy that it takes to start a chemical reaction, so the amount of energy to break and form new chemical bonds. So I showed you this in the last video. So these markers, it takes energy for me to break them apart. To break and form chemical bonds, same thing. It takes a certain amount of energy to break chemical bonds as well. Okay, so enzymes are going to be very specific to the types of chemical reactions that they can actually speed up. So if you talk about all the different types of chemical reactions that occur inside of your cells, there isn't one enzyme that works on all chemical reactions. A lot of the times, for each chemical reaction, a different enzyme needs to be used in order to speed it up. Okay? Now, the way an enzyme works, it only works with certain chemicals, and those chemicals that it works with are called substrates. Okay? So that's a new word, but you actually already know what substrates are. Substrates are reactants. It's what you start with at the beginning of a chemical reaction. So if we take a look at this example again, these are our reactants on the left. Arrow means a chemical reaction is taking place. Bonds are broken, new bonds are formed, and then these are our products over here. So when we're talking about enzymes, instead of calling these reactants, we call them substrates instead. Okay, so the substrates are just the chemicals that bind to enzymes, so that way the enzymes could speed up the chemical reaction. Okay, so if we take a look here, enzymes are very specific. They only bind to certain substrates. So we use the old lock and key example. Okay, now it's not a perfect analogy, but it definitely works in describing how the shape of the enzyme matches the shape of the substrate. So if we take a look at a key, okay, a key is very specific in shape, and the key that opens one door is probably not going to open another door. Okay, so if you took the key to your house and you put it into your lock at your front door, it's going to open that because the shape of the key fits the shape of the lock of your house. But if you take that same key and you go right next door to your neighbor's house and you try to put it in their lock and try to open up their house, it's not going to work because the shapes aren't exactly the same. And that's how enzymes work. There's all sorts of enzymes out there that can be used on different chemical reactions, but they have to match the shape of their substrate in order for them to work. So if we take a look at this for an example, okay, here's a picture, a really simplified picture of an enzyme matching its substrate. Okay, so the shape of the enzyme matches the shape of the substrate, and enzymes will only work on very specific substrates. They're not going to work on all chemicals, only very specific substrates. So if we go back to this example here again, okay, here's an enzyme. There's a spot in it where it's a certain shape, so the enzyme's designed a certain shape. This is called the active site. This is where the substrates are going to bind right here. The chemicals that are going to have bonds broken and new bonds formed, those are the reactants, or like I said earlier, when we are talking about enzymes, we call them substrates. Substrates will bind to the active site here. Bonds will be broken, new bonds will be formed, and the chemical reaction has occurred. Okay. All right. Now, how exactly do enzymes lower activation energy? So how do they make it to where you don't need as much energy in order to break and form new bonds? Okay, so here's basically how it works. I'm going to go back to using my markers as an example. So these markers take a certain amount of energy for me to break these apart. This time though, I'm going to say my hand is an enzyme here. Okay, so my hand is an enzyme. Enzyme binds to its substrate. When it binds to the substrate, it changes shape just a little bit. Okay, so when the enzyme changes shape just a little bit, it actually weakens the chemical bonds. So now it takes less energy to break the bonds and form new bonds. Okay, so one more time. Enzyme binds to its substrate. When it binds to its substrate, changes shape just a little bit. That slight change of shape weakens the chemical bonds and makes it easier to break them apart. Okay? So that's how an enzyme makes it so you don't need as much energy to break apart the chemical bonds. Now, as a chemical reaction occurs, the enzyme will remain bound to its substrates until the chemical reaction is done. So until the bonds are broken, until new bonds are formed. And when the chemical reaction is done, the substrates have been changed. They're no longer the same thing anymore. The enzyme, however, hasn't changed at all. This enzyme can be used over and over and over and over and over again on other chemical reactions. So you don't have to make a new enzyme every single time it goes through a chemical reaction. It can be used many, many, many times over and over again to do the chemical reactions. Okay. All right. Now, in order for enzymes to work properly and speed up a chemical reaction, they have to have certain conditions. So the first is a pH. pH has to be just right. Now, in humans, a pH of 7 is what is needed for most of our enzymes to work properly in most of our body. Some enzymes, though, in our body need a more acidic pH. So, for example, the enzymes in our stomach aren't going to work very well at a pH of 7. They're going to need a lower pH. So pH of 7 won't work for those enzymes. So all enzymes, they work best at a specific pH. Temperature is also important. So most enzymes will only work at a certain temperature. If the temperature is too high or too low, the enzyme will not work. So in humans, 98.6 degrees Fahrenheit is the best for our enzymes to work. And then substrate concentration also plays a role. So the amount of substrate actually present will affect how well an enzyme can actually do its job. Now, going back to pH and temperature, if the pH is too high or too low, or if the temperature is too high or too low, that causes the enzyme to change shape. Once it changes shape, it's not going to revert back. Okay, so it's basically what we call denatured. It's not going to work anymore. Okay, so one thing we could say here is the lock no longer fits the key. So if the enzyme changes shape, the substrates can't bind to it, and if the substrates can't bind to it, the enzyme can't speed up the chemical reaction. So let's go ahead and take a look at this internet example here. Okay, so this is kind of sum up everything we learned about enzymes so far. So here we have two chemicals, two substrates, reactants, what we're going to start with, and then here's three different enzymes. So here's one enzyme, two enzymes, three enzymes. This first enzyme, if you look at its shape, it has a shape that matches the carbon dioxide, but it doesn't have a shape that matches the water. This next enzyme has a shape that matches the water right here, but the shape here doesn't match the carbon dioxide. This enzyme has a shape that matches the carbon dioxide and a shape that matches the water. So this enzyme can be used in this chemical reaction to break the bonds in the carbon dioxide and form new bonds. Alright, so let's go ahead and take a look at this chemical reaction as it would occur without an enzyme. Okay, so the chemical reaction starts to take place. Bonds are broken, new bonds are formed, and in this amount of time, we made 1,000 products. Okay, so 1,000 of these carbonic acids. So remember, we started with carbon dioxide and water. Now we have carbonic acid, a new product here. Well, let's see what happens when we add an enzyme. So I add my enzyme to the reaction now. The water and carbon dioxide will bond to the enzyme, speeds up the chemical reaction, breaks bonds, form new bonds. In the same amount of time that it took to make 1,000 of the product last time, now we were able to make 107,000 in the exact same amount of time. Okay, so the enzyme made it so you needed less energy to do that, and it sped up the chemical reaction so much faster. It made it occur so much faster. Okay, well, towards the end of the notes there, I talked about the fact that if you have a pH too high or too low, enzymes don't work properly because their shape changes. So I'm going to go ahead and lower the pH here, and let's see what happens to this enzyme and to the chemical reaction. Okay, here you can see the enzyme change shape. These substrates no longer fit the enzyme. So the chemical reaction can occur, but it's going to happen at a much slower rate. So we're only going to make 1,000 of the product here. Okay, so instead of making 107,000, we're back to only making 1,000. Okay. All right, another example here. So if you take a look, go ahead, take a second, choose which one you think is going to be the enzyme that these substrates match. Okay. All right, you should have chosen the middle one, the red one here. All right, let's take a look. We have 2 hydrogen peroxide molecules, and the chemical reaction is going to occur. Bonds are going to be broken, new bonds are going to be formed, and we're going to make our products. So in that amount of time, we made 1,000 of each of the products here. Let's add our enzyme and see what our enzyme does to the amount of products we can make. Okay, so substrates bind to the enzyme, bonds are broken, new bonds are formed. In the same amount of time it took to make 1,000, we are now able to make 5,600,000. So that's a huge, huge difference, going from 1,000 to 5,600,000. So enzymes really make it so that you can speed up the chemical reactions. You can make more products faster. All right, now let's change the temperature. Okay. So if someone is really sick and they have a high fever for a very long period of time, that can actually be a really bad thing. In fact, it happens all the time where people can actually die from these high fevers. And one reason for this is the fact that when you have a higher fever, so I'm going to raise the temperature here. Okay, when you raise the temperature, it causes our enzymes to change shape. So temperature is higher, enzyme changes shape. These substrates will no longer be able to bind to the enzyme, so the enzyme can't speed up the chemical reaction, so the chemical reaction occurs slower again. So instead of making 5,600,000 of the products, you're back to only making 1,000 each. Okay, so that is a huge, huge difference. So if someone has a high fever for an extended period of time, their enzymes change shape and they can't do their job to keep those chemical reactions going. And if you remember, if the chemical reactions in your cells stop, your cells are going to die. If your cells die, you die. Okay. Now, same thing will happen in the opposite case if your body temperature drops. If your body temperature drops too low. So in that case, same thing's going to happen. The enzyme's going to change shape. The substrates won't fit, and you can't speed up the chemical reactions. Your chemical reactions stop or slow down, and your cells begin to die. Okay. All right. So that is enzymes and how they speed up chemical reactions. Yeah.